So while I'm waiting for my slides to load, I can just tell you a bit about my lab's research program. So we've already heard about how cross-species approaches can be handled or assessed using different types of transcriptional atlases, how you can compare and contrast gene expression products across, say, rodent, C. elegans, and human models. My lab is interested in leveraging these same types of approaches, but in this case going after the genetic or molecular processes that might bias the formation of large-scale networks in the human brain, with the hope of applying these approaches to study vulnerability factors that could lead to the onset of psychiatric illnesses. Now, you may have noticed this from the first slide. The title of this talk is a bit different than the one on the program. In part, that's because my graduate student, Kevin Anderson, is a bit too successful. He initially put in for a poster session, and then he was given the opportunity to present that work in the form of a talk. That was what I was initially planning on presenting here today. So if you're interested in the abstract and title from the symposium today, I highly, highly encourage you to check out Kevin's talk tomorrow. In this talk, he's going to discuss how the presence of different levels of somatostatin or parvalbumin inhibitory interneurons could have effects on local sort of patch firing rates at the level of cortex, how you can account for heritability in brain function through gene sets derived off of these markers, and how that can be understood or yield information on the genetic bases of schizophrenia. So Kevin's going to present that tomorrow. Instead, what I'm going to present today is an extension of that initial line of research. Just to give you some key takeaways, because these talks tend to sort of fly by like a whirlwind, I'm going to leave you with 3 key points. I'm going to hope to convince you that there's a subtle but stable signature for behaviors and factors that are associated with the presence of major depressive disorder, that these signatures are stable not just in structural estimates but also functional estimates of brain function. To do this, we're going to use 3 independent data sets. So the UK Biobank, the Harvard Brain Genomic Superstruct Project, and then data from the Enigma MDD Consortia. I'm going to suggest to you that these stable profiles of brain anatomy and function converge on evidence that implicates somatostatin inhibitory interneurons as a key basis for a biological factor that could underlie vulnerability for the onset of depression. And I'm going to suggest to you that these in vivo estimates of cell types that are implicated in depression are consistent with data sets we see when we look at ex vivo analyses of post-mortem brain tissue. So brain tissue from patients that have passed away, donated their brains to science, and then we can sample. So as a field, we've had some really elegant models of how shifts in brain anatomy and function might relate to vulnerabilities for the onset or relate to the maintenance of psychiatric illnesses like depression. So this is one of my favorite figures ever in I think all of neuroscience. So this is from a 1949 review by Paul MacLean of the limbic system. Even in this early figure, you can see sensory information streaming into the hippocampal formation. You can see it traveling down the spinal cord related to freezing responses. You can see MacLean already suggested that medial prefrontal cortex, for instance, might be involved in affective functions such as guilt. So we knew a lot about how the brain functions, at least at the theoretical level, way back in sort of the early times of Paul MacLean. Now, we've obviously expanded and refined these theories since that point. So in my area, here's the classic theory that I grew up with, which is Helen Mayberg's theory for how depression emerges and is maintained in the brain. So here you see she's expanded the scope of the model. Now it incorporates huge swaths of cortex, each with a circumscribed function. Once again, the medial prefrontal cortex comes up as a sort of key node in that, sort of mitigating between sort of somatic and vegetative deficits and executive functioning processes. So we've had these really elegant models for a long time. However, as a field, we've struggled mightily to try to find actual empirical data sets that support the basis of these models. And so there's been some notable successes, right? So the paper, which I love, I think most of all, is this Wayne Drevitt's paper from 1997. So in this, he demonstrated there's altered metabolic function in patients with unipolar and bipolar depression and reductions in prefrontal gray matter in associated territories. These are the same territories that were implicated by MacLean and Helen Mayberg and others. However, it seems that as a field, when we try to establish the reliability of these effects, it's almost like we're chasing smoke at times, and so we increase our sample sizes, and as we increase our sample sizes, the effect sizes we're hunting get smaller, and then we increase our sample sizes, and the effect sizes get smaller again. So just as one example, in 2012, we published a paper when I was on postdoc with Randy Buckner, using 1,050 individuals, basically recapitulating many of the findings from that earlier Wayne Drevitz discovery. Here the effect sizes are much, much more subtle. In this we looked at individual variability and behaviors related to depression, showing shifts in medial prefrontal thickness, tracking vulnerabilities for the onset of the disorder. We were pretty happy at the time. 1,000 participants is a huge study. Less than 4 years later, thanks to Paul Thompson and the Enigma Consortia, they were able to cobble together a much, much larger data set. So Small and colleagues published in 2016 this molecular psychiatry paper in over 10,000 individuals, demonstrating that you see many of the same regions popping up, but once again, the effect sizes are much smaller than one would hope, at least in terms of brain anatomy. So giant sample sizes, effect sizes are getting smaller, at least in the area of in vivo imaging, right? Or we're becoming more accurate in our understanding of the size of effects that we should expect. Now, in conjunction with this, there's been a whole series of incredibly elegant bench lab neuroscience, post-mortem tissue style analyses, looking at animal models and case control models that are implicating some really important cellular or molecular processes that may contribute to vulnerability for the onset of depression. So today I'll be talking about somatostatin inhibitory interneurons. So there's prior evidence, at least in post-mortem tissue samples, to suggest that there's a reduction in the expression of somatostatin in patients who have passed away, suggesting it may be implicated in the pathology of the illness. Now, excitingly, if you take that same product and look at it in animal models, if you activate somatostatin inhibitory interneurons, you see a rescue in at least animal models of depression. So rodents, for instance, will perform better in forced swim tests. They don't display as much learned helplessness, so they're much more likely to escape when escapes are available, for instance. So it looks like there's a clear biological process that we should be able to target if we're looking at the pathophysiology of depression. And so what Kevin wanted to do in this study is see if we can merge these two types of data sets together. Right? Given that there's large, high-powered in vivo imaging data sets with genetic data available, so data sets like the UK Biobank, we have post-mortem tissue banks that are becoming increasingly available. So in this case, we're using the Allen Institute data set. Can we find converging evidence across these distinct modalities to implicate a particular class of cells or a particular molecular process associated with vulnerability for the onset of depression? And so this is leveraging both small, sort of relatively small-scale ex vivo studies of gene expression and trying to couple it with these high throughput, massive in vivo imaging collections. So to start, we used the Smith 2013 definition for depression from the UK Biobank. So in this paper, they defined a single episode, moderate or severe depressed patients. And so here you can see the proportion of those samples. Here you can see the proportion of those samples within the UK Biobank dataset. Since we in my lab tend not to trust one single measure to know we're looking at a phenotype, we wanted to check those against self-reported neuroticism. So this is the, I think, subscale in neuroticism that's available in the UK Biobank. As expected, you see as the severity of recurrent depression increases, you see an increase in neuroticism. Arena already mentioned the polygene approach. So this is the high 2016 genome-wide analysis for depression. Here we're showing that there's increased polygenic vulnerability for depression in individuals that are at least self-reporting increased rates of depressive episodes. Once again, suggesting that since this is a population study, one's never entirely sure how good the data is, but it suggests converging evidence across multiple assessment modalities to suggest we're hitting something that may be consistent with major depressive disorder. Now, given the fact that the single episode reports in the UK Biobank are a bit unstable from time point to time point, we collapsed our data sets to look at moderate and severe depression against individuals who don't report a lifetime history of a single episode. To start, we looked at estimates of cortical thickness. So this is using the FreeSurfer pipeline. Unlike a traditional free surfer analysis, we applied the Schaefer Atlas, Thomas Yo's lab's recent functional network atlas to our data. So those are the parcels from the Schaefer Atlas. And we looked at the relationship between moderate and severe depression and health in terms of cortical thickness. And so you note what we observed is consistent with prior reports. We see a preferential reduction in medial prefrontal thickness in the left hemisphere in individuals that have a probable diagnosis of depression in the UK biobank. So it looks like a subtle effect, but at least if you squint, it's consistent with stuff in the prior literature. Now to check how somatostatin might be associated with this profile, what Kevin did is he took the Allen Institute data set, took individual samples from participants, normalized expression for each gene across cortex, and then averaged the expression of each gene within a parcel in the Schaefer Atlas, and then looked at the relationship between somatostatin expression in each of those individual parcels across the entire cortex, and assessed how well that correlated with the effects that we're seeing related to major depressive severity in the UK biobank data set. And so what we observed was an effect where there's increased thinning in participants with heightened levels of depressive symptomatology. That's where we see increased expression of somatostatin in the Allen Institute data set. So where there's thicker cortex in depression, those are areas that are less rich in somatostatin. Where there's thinning associated with depression, those are areas where there's preferential expression of somatostatin. Now, one thing you always want to check when you do these types of analyses is assess if this is preferential to somatostatin in any kind of way. So Kevin reran those analyses across every single gene that's expressed within cortex, looking at its relationship to the map we have of depression, and show that this is a preferential relationship between that one single marker for somatostatin inhibitory interneurons and that map we have of associated with the presence of moderate or severe depression. Now, this is one way to look at the relationship between somatostatin and inhibitory interneurons. You can focus on a single marker. However, for folks in the area, you know that cells don't only express one marker. So there's many types of inhibitory interneurons. They each have complex expression profiles. In order to tackle this in a sort of more systematic way, Kevin used data from Lake and colleagues' 2018 paper and applied this amazing new approach from Aaron Newman that came out recently this year called CyberSort X, which was originally developed to do cell typing within tumors. Kevin adapted it to look at cell typing within the human brain. And so you can take the 24 different cell types from Lake and colleagues and then use the CyberSort X program or analysis pipeline to see how those cell types are distributed across cortex. When Kevin did this, he noticed that there's a preferential relationship between thinning in regions associated with depression and increased presence of somatostatin inhibitory interneurons in the Allen Institute data set. So here you see the two subtypes of somatostatin neurons. Inhibitory neuron 7 is a superficial a somatostatin interneuron that appears in superficial layers of cortex. Inhibitory neuron 8 is a deeper layer of somatostatin neurons, suggesting that there's a converging evidence both through single marker and cell deconvolution approaches that are implicating somatostatin neurons in the anatomical basis of depression. Now I mentioned we want to show these are consistent. So one way to do this is to look at existing data sets. So this is off of the Enigma Consortium data set. Here you see it's consistent with profile we see in the UK biobank sample. This is using the Desigan Atlas data from the Enigma Consortium. So the effect sizes are consistent across our data types. And the Enigma Consortium also implicates somatostatin through that same approach that I just referenced, suggesting there's some preferential relationship between somatostatin and these depressive phenotypes. You can also do this with functional correlates of depression. So this is looking at resting state amplitude fluctuations. This is a standard deviation of the bold signal across a resting state run. So for this we used Thomas Yo's CBIG pipeline to compute our resting state amplitude fluctuations. Really interesting, what we observe is a increase in the standard deviation of the bold signal in association cortex relative to unimotor cortex. An increase in the RSFA fluctuations is what's been hypothesized by some elegant rodent models looking at distribution of somatostatin neurons across cortex. So as they become more dysregulated or less prevalent, you should see an increase in amplitude fluctuations. Consistent with this, we see in the Allen Institute data set those resting state amplitude fluctuations, or the relationship between those fluctuations and major depressive disorder, also tracks the presence of somatostatin in the Allen Institute data set. Once again, this works both if we look at cell types or the individual expression profiles. Now, this replicates, so we can look in the genome superstruct data set. Here we're not looking at case control, we're looking at individual differences in negative affect. So it's consistent across both data sets, also implicating somatostatin inhibitory interneurons. You notice this effect is not that strong. Genome superstruct is a relatively young sample, so if we reduce the sample down to just those individuals over the age of 24, so to more closely, at least as close as we can match the biobank data set, we see an increase in the size of that effect. Once again, it seems to be preferential to this class of inhibitory interneurons. Two last data points I'll show you. You can also, instead of doing amplitude fluctuations, calculate mean parcel connectivity in resting state data to get a pointwise estimate at each parcel. If you do this, you see that same effect where there's reduced global connectivity within a parcel. We also see increased prevalence of somatostatin inhibitory interneurons. Now, I mentioned ex vivo post-mortem analyses. So we took data from the Gandel Science paper, where they have 83 patients with depression and 86 healthy comparison individuals. So these are post-mortem tissue donations from a patient sample. If you look at the expression or the deconvolution of cell types within that post-mortem tissue, you see it lines up really nicely with the estimates we're getting from the Allen Institute data set based on the spatial profiles we're seeing related to depression. Once again, deep layer somatostatin inhibitory interneurons seem to be preferentially relevant to the pathophysiology of depression. So I'll stop there. I want to thank all my collaborators on this project, in particular Kevin, who should be finishing up grad school soon, and the folks in Thomas Yo's lab who helped implement the CBIG pipeline.